It's a pleasure to introduce Chuba as thesis student today. It's been a pleasure to mentor him for the last few years. And it's also really an honor to have you guys here to listen to this seminar. In particular, I'd like to welcome his parents who came all the way from Nigeria to be with him today. So afterwards, please introduce yourselves to his parents. So the fundamental question that Chuba has really been focused on is, in a population of cells, how individual are the single cells from one another? Are they exactly the same, or are they different from one another? And this is a question, it seems very simple, but it's an unknown question, and it crosses all of biology and medicine. And this question of how similar or different are cells within a population. And it's particularly important for regenerative medicine, because we really need to know when we're differentiating cell types into specific lineages like heart and neurons and muscle, whether the cells within those culture regimes are the same, or whether there's all kinds of different cells within those populations. Because you certainly wouldn't want cells being put back into patients that have a mixture of cell types within them. And so Chuba's really been focusing on this question, which we think is a really important one, not just for regenerative medicine, but broadly for biology. And so he's going to tell you about these experiments that he's been working on over the past couple of years, which I really think have led into some really interesting avenues. So without further ado, Chuba.
Good morning. I'm Chuba and today I'll be talking about some of the work I've done towards the development of novel approaches with which to visualize cell-specific gene expression patterns. Parkinson's, leukemia, diabetes. These are all conditions to which the vast majority of us in this room are susceptible to, by virtue of the fact that we're human. So it is very important for the people in the know, people who study these kind of conditions, to develop methods with which to either prevent the onset of such conditions in the first place, or to improve the quality of life for the unfortunate amongst us that are already afflicted. Now, all three of these conditions listed here, to a very large extent, are as a result of damaged or malfunctioning cells. So the logical, or not necessarily simple solution to this problem might be to replace these damaged cells in vivo. The question that automatically arises from that is, how do you replace damaged cells in vivo? There's a very high likelihood that most of us in this room, at one point in time or another, have heard of human embryonic stem cells, or stem cells in general. Human embryonic stem cells are a remarkable class of cells because they have the unique ability, through their pluripotency, to give rise to a number of cells in the human body. This immense power vested in human embryonic stem cells makes them a prime candidate for a possible source of therapeutically relevant cells with which to replace this damaged tissue in vivo. So how do we exploit the potential of this great resource known as the stem cell? There are lots of valid and varying opinions out there, two of which are listed on this slide. We feel we must have great differentiation protocols with which to drive towards different cell types that we hope are therapeutically relevant. And second, please remember that since human embryonic stem cells are pluripotent, it would be nice to have a way to tell the difference between the different cell types that are emerging. Keeping these key points in mind, we can then ask ourselves some specific scientific questions. Can we examine the nature of complex cellular populations and discover biomarkers from them? Or can we create tools to verify the identity of cells obtained from stem cell differentiation protocols? Let's look at the first question in a little bit more depth. Can we examine the nature of complex cellular populations and discover biomarkers from them, with which to hopefully distinguish distinct cell groups from the complex population of cells? A lot of great work has been done towards the creation of differentiation protocols with which to drive the human embryonic stem cell to stem cell derivatives of therapeutic relevance. However, what remains unanswered, what remains not that well understood, is the degree of homogeneity or heterogeneity within the individual members of the stem cell derivative. To that end, we asked ourselves this question: are single cells that have been isolated from a fairly heterogeneous group of stem cell derivatives, based on the expression of a unique cell surface marker, completely identical to one another? To answer this question or this riddle, we needed a cell type of choice to work with. We chose definitive endoderm. It is a primary germ layer that gives rise to very important organs in the body, organs like the lung, the liver, and pancreas. To obtain the definitive endoderm cells, we subjected human embryonic stem cells to 5 days of activin treatment in limiting concentrations of fetal bovine serum. After 5 days of this exercise, we were able to obtain definitive endoderm cells in a tissue culture dish. Going a step further, the subgroup within the resulting definitive endoderm cell population that expressed the unique cell surface marker CXCR4 on their surface were isolated as single cells within single wells of the 96-well plate. Now, since we're basing the degree of similarity or difference between our single definitive endoderm cells on the unique patterns of gene expression found within them, one of our major tasks was to identify genes that are consistently and considerably expressed in the definitive endoderm cell type. To this end, and in collaboration with some of my colleagues in the lab, we were able to perform 2 key experiments on our definitive endoderm cells. The first one is an exon array experiment. The second one is the sequencing of the RNA content within the cells. From the intersection of these 2 experiments, we're able to select 22 endoderm-specific genes. To this list, we added SOX1, which is an ectodermal marker, as a negative control, and glyceraldehyde 3-phosphate, which is a housekeeping gene, as a positive control. In order to obtain the patterns of gene expression present within each single cell, we use the technology seen on this slide. It is a biomark gene expression array. The 48 wells you see to the left-hand side of this little microfluidic chip serve as deposits for genetic material extracted from single cells. Whereas the 48 wells you see to the right-hand side of the microfluidic chip represent deposits for primers that have been designed to represent genes of interest. Intelligent plumbing on a microfluidic scale facilitates the mixing of the genetic content from the cells with the primers that have been designed to represent the genes of interest. This culminates in over 2,000 quantitative real-time polymerase chain reactions occurring on the same chip. Using the biomark technology that we showed on the previous slide, we were then able to elucidate the patterns of gene expression present within single definitive endoderm cells. There are 40-odd traces on this slide, and each one of these traces represent the unique patterns of endoderm-specific gene expression seen within our single CXCR4 positive definitive endoderm cells. And as you can see, there's a high degree of heterogeneity from cell to cell. To further accentuate the individuality of each individual CXCR4 positive cell, we designed and developed a software tool called SC Express. It basically takes the gene expression patterns from the biomark system and converts them into 3-dimensional shapes in their stead. The details of the methodology through which this is accomplished will be explained shortly. So in order to generate each one of these cell-specific shapes, an input data matrix of the following form is required. In the first column, we have fold enrichment values that have been calculated for each of the genes of interest. Whereas in the second column, we have cycle threshold values that have also been recorded for these genes. Now please note that the fold enrichment values and the cycle threshold values serve as indirect measures of the quantity of each one of these genes expressed within the cell. It is also possible to obtain both values using the biomarker gene expression arrays that we showed on the slide 5. The first step in the creation of each cell-specific shape is the generation of a pinwheel on the XY plane. The number of spokes within each wheel is exactly equal to the number of genes under consideration. In this case, 24, but that number can be scaled up or down to suit any user's particular need. On the left-hand side, you see the pinwheel in free three-dimensional space, whereas on the right-hand side, you're looking at the same wheel, except from a bird's-eye perspective, or a plan view. The next step in the creation of each cell-specific shape is the generation of an array of stems in three-dimensional space. To understand exactly how this comes about, let's take the example of the gene listed below, gene 1. First, we locate the spoke within the wheel that represents gene 1. And we count a number of units outwards from the center of the pinwheel on that spoke. The number of units we count outwards is exactly equal to the cycle threshold value recorded for gene 1. Now we can mark that spot. Next, the base of the stem is placed on that spot. The stem rises above the horizontal at a 90-degree angle to the XY plane. The height of the resulting stem is equal to the fold enrichment value that has been calculated for gene 1. Now, with a little bit of thought and imagination, I think we can all start to see how an array of stems will arise in three-dimensional space, as this procedure is carried out for every single gene under consideration. Please note that the resulting array of stems serves as a skeletal framework upon which the eventual three-dimensional shape is constructed. As a matter of fact, connecting the tips of all the stems results in a three-dimensional shape that note has been created after careful consideration of the gene expression patterns present within that cell. I think the next and perhaps most logical question to ask at this point is, how reproducible are these shapes? That is to say that given two aliquots of the genetic material from the same exact cell, can you reliably and consistently recreate the same three-dimensional shape? Panels A and B on this slide are three-dimensional shapes that have been created from a single CXCR4 positive definitive endoderm cell at two different instances. While C and D are also three-dimensional shapes that have been created from another cell at two different instances as well. And as you can see, there's a fair bit of resemblance between the shapes. Now, as I mentioned earlier, we designed SC Express to convert the gene expression patterns present within a single cell into three-dimensional shapes that can be represented. And I would like to show you a brief demonstration of the actual program now. SC Express was designed in a popular programming tool that most engineers use. To get the program SC Express to work, all you need to do is type in the name of the program. To prompt you for some input data, we've created two files. This first one contains data from a pilot experiment done on definitive endoderm cells. Now, this second file contains data from a technical replicate of that experiment. Basically the same experiment, just performed on a different day in slightly different conditions. Upon data entry, SC Express checks the format of the files to make sure it agrees with what it expects. If that checks out, it comes up with a graphical user interface of the following form. Please pay attention to two key prominent things in this group. There's one visualization window, VW1, and its accompanying buttons. And there's VW2, and its accompanying buttons as well. The manner in which I've entered the data will cause the three-dimensional shapes from the pilot experiment to be shown in VW1, while the three-dimensional shapes from the technical replicas of that experiment will be shown in VW2. So in order to see each one of the cell-specific shapes, all you need to do is select the cell from the drop-down menu, pick cell 1. Click on cell-specific 3D, and the shape shows up in the allotted window. Using most of the tools that are available to any MATLAB user, you can zoom in on the shape and move it around where you like. And if you wanted to return back to its original vantage point, you click on cell-specific 3D, and back it goes. So this is what cell 1 looks like in the initial or pilot experiment. Let's go ahead and compare that to what cell 1 looks like in the technical replicate of that experiment. That's what VW2 is for. You can select the cell, cell 1, click on cell-specific 3D, and the shape shows up. As you can see, there's a fair bit of resemblance between the shapes. But this is just one cell. You can try this for other cells in the list as well. Let's try something for maybe further down the list. You click on cell. So this is what cell 30 looks like in the initial or pilot experiment. You can compare that to cell 30 from the technical replicate of that experiment. And as you can see, there's a fair bit of resemblance between these shapes as well. Using SC Express, we're then able to generate cell-specific shapes to represent individual CXCR4 positive definitive endoderm cells. And as you can see, the shapes are rather different from one another, basically corroborating what you saw shown on a couple of slides prior with more traditional methods. Our next challenge was to see if we could find biomarkers with which to distinguish distinct cell groups from one another. So today, cell-specific gene expression patterns are often displayed using heat maps from microarrays that you can all see on the left-hand side. We wanted to develop a complementary method to that, a method in which, with just the expression of a few biomarkers, you could visually distinguish between cells of different types. Since we already have a protocol with which to derive definitive endoderm cells from human embryonic stem cells, we chose to start by seeing if we could find biomarkers with which to tell the human embryonic stem cells apart from the definitive endoderm cells that were derived from them. Now please note that even though we derived the definitive endoderm cells from the human embryonic stem cells, the genes that are prominently expressed in human embryonic stem cells aren't necessarily those that are prominently expressed in definitive endoderm, and vice versa. Thus, the first thing to settle on was a class of genes that are prominently expressed in the human embryonic stem cells, in the definitive endoderm cells, and in a broader array of cell types in the event that we wanted to make this method extendable to touch other cell types as well. We settled on the class of housekeeping genes because numerous experiments have shown that these genes are expressed in a multitude of cell types. Using the biomark gene expression arrays, we're able to analyze the gene expression patterns of the housekeeping genes within definitive endoderm cells, and analyze the expression of the housekeeping genes within human embryonic stem cells. Each one of the traces on this slide also represent the unique patterns of gene expression found within a single endoderm cell, in this case, or human embryonic stem cell. And as you can see, patterns of gene expression are fairly homogeneous from cell to cell. To put this in contrast, let's compare the gene expression patterns of the endoderm-specific genes within the single definitive endoderm cell to the housekeeping genes in the same exact cell type. As you can see, housekeeping gene expression is much more consistent from cell to cell than is endoderm-specific gene expression. That's why we chose this class of genes as a possible source of biomarkers. So in order to find the main axes of variation within our data set, we perform principal component analysis. It is a mathematical method that can tell you the degree of variation present within a particular data set. Or another way to think about that is that it takes the number of possibly correlated variables and converts them into uncorrelated variables or principal components. For me, usually that definition is a little too tough. So I try to break it down into an analogy so that I can understand it. So take, for instance, all of us in this room. We can think about variables with which to tell each and every single one of us apart from the other one. Some of these variables are hair color, hair length, height, nationality, skin color, and so on and so forth. Now, even though in all our minds, these are good variables with which to tell each person apart from the other one, you start to notice that some of these variables are actually correlated to one another. For instance, if you're as dark-skinned as me in this room, you're probably Nigerian. And if your hair is long, you're probably female. This is what principal component analysis does. It basically takes these variables that are essentially correlated with one another and collapses them into a single principal component. This is to make sure that all the axes of variation within the data set are completely orthogonal to one another. So we perform principal component analysis on our human embryonic stem cells and definitive endodermal cells. Plotting the two most statistically significant principal components, principal component 1 and 2, we're able to discern visual separation between the human embryonic stem cells and the definitive endodermal cells that we have derived from them. Seeing this result inspired the addition of more cell lines to the system. We wanted to see if even more cell-specific clusters emerge with the profiling of this housekeeping gene set. To this end, we added the following cell lines to our list. Induced pluripotent stem cells that have been derived from human dermal fibroblasts, definitive endodermal cells that have been derived from the induced pluripotent stem cells, and the ENCODE sanctioned FG2 and 29P2 cells. Plotting the resulting principal components that came from this more extensive data set, cell-specific clusters arose as well. So we can see a unique cluster for the human embryonic stem cells, here and here, one for the induced pluripotent stem cells, one for the HEPG2 cells, and so on and so forth. Now, as I mentioned earlier, one of the main reasons for performing the principal component analysis is to discover the major axes of variation within the data set. We can take further advantage of this though, by identifying the actual variables within the data set that are most responsible for the variation you're seeing. Along these same lines, we looked even deeper into our data set and were able to discover 3 genes that accounted for most of the variation that we saw. These 3 genes are NONO, or non-POU domain containing optima, ACTB, or actin beta, and LDHA, or lactate dehydrogenase A. At this point, we tried something a little different. We used the gene expression levels of all 3 genes as X, Y, and Z coordinates in the Cartesian system, you can see. And we went ahead and plotted all the cell lines on the same point. And to our surprise, cell-specific clusters arose. That's a unique cluster for the HEPG2 cells, one for the iPSNU cells, one for the human embryonic stem cells, the endoderm derived from the human embryonic stem cells, and so on and so forth. As a control to this experiment, we picked 3 other genes at random from our housekeeping gene set and did the same thing. We used them as X, Y, and Z coordinates in the Cartesian coordinate system as well. And as you can see, the cell-specific clustering that we saw on the slide prior was lost. So up until this point, we've shown that single definitive endodermal cells showcase individualistic patterns of gene expression. We've also been able to show that housekeeping genes show promise as a potential source of biomarkers with which to isolate distinct cells from a complex mixture or population of cells. We turn next to this question. can we create tools which to help verify the identity of cells obtained from stem cell differentiation protocols? The eventual dream or goal in this case will be to build a database in which the unique characteristics of each cell can be quantified and converted into a cell-specific shape using some transfer function that we refer to now as H of omega. We hope that such a database will eventually come to serve as a point of reference for scientists who work extremely hard to perfect their own differentiation protocols. In more detail, scientists could apply their differentiation protocols to human embryonic stem cells in search of unique cell types of therapeutic relevance. They could then create cell-specific shapes to represent those cells as well. In the event that the cell-specific shapes created to represent the experimental cell types are in close accordance or close similarity with those of the somatic cells, we hope that this will eventually come to be accepted as a means with which to validate differentiation protocols. Now, I hope the question that's most preeminent on all our minds right now is, what is the transfer function H of omega? What does it mean? Turns out that H of omega is basically SC expressed that has been expanded to work for multiple cell types as well. On this slide, we have two panels, A and B. These are shapes that are created to represent a definitive endoderm cell population this time, as opposed to a single definitive endoderm cell. And this was done at two different instances, A and B. As you can see, there's a fair bit of similarity between these shapes. So we could refer to this maybe as the shape or the face of the definitive endoderm cell population. In summary, the work we've shown here suggests that it's possible to discern differences between cells of the same type. We've also been able to identify housekeeping gene biomarkers that allow for visual distinction between a number of different cell lines. And we've shown what we hope here is the inception, the computational tool with which to aid the creation of differentiation protocols. As the fields of stem cell engineering and cell replacement therapy take sure and steady steps towards becoming a reality, the ability to isolate pure populations of therapeutically relevant cells is becoming an even more pressing need. As I've tried to show here with the work I've been fortunate enough to do over the last 5 years of my life, I've always tried to take steps towards fulfilling that need. Over the years, I've come to understand life as being very analogous to a game of soccer. Teamwork is required, unending challenges. No matter how good you are as a person, you need good people around you to help you too. And the best players on any team are those that make their teammates better. I'd like to thank the following people on this slide for definitely making me a better man and a better scientist. My parents, who came across the Atlantic to see this today. Nice to see you. Thank you very much. All the members of the Data Lab have been very, very generous with their time and their attention. The members of the Quick Lab that introduced me to the single-cell gene expression experiments in the first place. Fuad in the Tang Lab, who's responsible for a lot of the computational stuff we're able to do. From my parent department, for Saltman, who's our chair. Al-Daisro, who's my academic advisor. Rebecca Farron, who's my chair today for the Defense Committee. My good friends over Lydia and Christine. From the genetics department, Dr. Gavin Sherlock was answered a lot of questions from me over the years. Michael Snyder, who supplied a lot of the cell lines we're able to use for our experiments. My good friend Ray, Truti, Maria, and the list goes on and on. It's necessary. From the Clark Lab, I'd like to thank Piero D'Alebra, who was instrumental in the isolation of single CSCR4 positive endoderm cells. And from the FACS facility, Catherine. From my friends at Illumina, Brian Rapkin, Michael Zerline, Mark Pratt, and Aidan Lee, and this person. I can't tell you how much it means to me to see so many familiar faces in the audience so early in the morning. Thank you very much for making it. I really appreciate your time, your attention, and your patience through the long talk. If you have questions, I'll try to listen.
So, do you have an idea of how sensitive these cell shapes are to a specific cell line? So, for example, if you're trying to make a specific population, if you have 10% of more of one gene, does that completely change the cell line?
The shape that represents the cell line?
Mm-hmm.
Yeah. So as you saw from the wheel, there are different axes, right? So there's 24 in this case. But as you expand the number of axes that you use, the less the difference in one gene matters. That makes sense. So you see what I'm saying?
Mm-hmm.
Yeah.
Yeah, so one of the last pictures you showed was a distinct shape for a cell population. And I was wondering, like, for instance, the genetic endoderm sharks, how different would the population shape be from a single endoderm cell?
So I think that's a good question. Because when you look at enough of those shapes for the single definitive endoderm cells, and you look at the one that comes from the population, it starts to look almost like an average. And every once in a while, you have an outlier, right? That doesn't really fit the rest of it. But after a while, it starts to look like almost the eventual shape of the population starts to look like an average of what you sense. Fine.
Yes, you showed these 3D shapes to sometimes compare and see how similar to different cells are. My question is, do you have a way to quantize or express error?
Yeah.
And I'm wondering how you determine the threshold for when things were similar enough.
Right. So in this wheel, right? In each wheel, so say we're comparing cell 1. So this is cell 1. Can you see this? And this is cell 2. Now they all have the exact same axes, right? So for gene 2, there'll be some stem rising above the XY plane, right? That we show. Now for gene 2, in this case, there'll also be some stem rising above the XY plane. Let's call this stem 2. Let's call this stem 1. What you can do is from the center of the pinwheel, you can draw a vector to the tip of that stem. And you can do the same thing here as well. Now note that this vector has two properties, right? Magnitude and also the direction in which it points. So there's an angle of this vector to the actual horizontal, we'll call that theta 2 in this case. And we'll call this angle theta 1 in this case. Now to find the similarity between each vector, call this vector A1, call this vector A2. Right? So if this cell is the same as this cell, these two vectors should be exactly the same. Right? And you can discover the difference between the vectors by taking the dot product, which is that A1 dot A2 is equal to the magnitude of A1 times the magnitude of A2 multiplied by the cosine of the angle between them. Right? So you know what A1 is, you know what the magnitudes are, you can solve for cosine theta. And as theta approaches 0, both vectors start pointing in the same direction. You will get the same. And
great talk. For your transfer function, is it unique between cells, cell lines?
no, it's the same thing for everything. It's the same thing for all cell lines that we use so far.
So could you predict what the last function, like the output is going to be of the experiment, since you know the transfer function?
Well, it depends on if we know the patterns of genes that are already within the cell. So a lot of people have done a lot of great work to figure out, the profile of cell types, figure out the genes that are most highly expressed in a particular cell type. And that's why such work is so great, because like you were saying now, you don't need to go and play with the cells anymore. You already know what the genes are. You already know what the transfer function is. They're like signals and systems, right? You can tell what the output is. So, yeah. Yes, please.
I have three practical questions. Number one, biomarker chips cost? Number two, how long does it take to obtain data? From the time you take a cell off the plate to, and then do you think undergrads could use your SC Express software?
So the first question, how much do the chips cost? $240 for one. If you buy 5, you get a $40 discount. Then the second question.
How long does it take, and could undergrads run the program?
3 hours. And yes, I think undergrads can run it. It's just like, it's a GUI, like OS 10 or Windows. It's just quick.
We think it might be useful in our new tissue engineering and biomaterials lab, or the agricultural and health cells, and have something to read out.
We'd love to talk to you. Hurry up.
Just a kind of statement from a layman here. It looks like your SC Express especially could really be the beginning of a wonderful tool for, even when it comes to writing grant applications, because of the fact that it's very visual, and for people who want to give money and that kind of stuff, it really says a lot. Most people, when you look at something and they see that, like you talked about how it's very reproducible, the different kinds of whatever cell expressions. So it looks like it could be very, very useful for researchers when you want to write a grant proposal, something like that, or even for private.
I have one more question. How do you know which cell is cell 2 or which cell is cell 30? Is that different plates or different wells?
Different wells. So the cells are isolated as single cells within 96 well plates. And you isolate each cell individually. You reverse transcribe the RNA to CD and amplify that.
So every well has a different set of growth factors, or how do you always get cell 30 in position 30, and how does it know it's cell 30?
Oh, so when you isolate the individual cells into a 96 well plate, I break down the cell membrane and split up the genetic material physically with a pipette into half. So I have 2 plates, and each plate has the genetic material of the same cell in the exact same well. I just split it in half to reproduce it. Does that make sense?
I still don't know how cell 30 knows it's cell 30. Are they exactly 30? You know what I mean?
Okay,
I don't understand.
So were you
embryo, I guess.
were you comparing the assay at 2 different times on the same cell?
Yes. That's why it's correct. Okay, so in each 96 well plate, you go through fluorescence activated cell sorting. There are different wells. And you're able to get this one. Let's call this one cell 1. Or maybe cell 30. In this well. But then what you can do is take the genetic material in this well and you can split it in half, right?
So it comes from a very particular region of the fat spot?
This is an actual physical
Is that
split.
what makes it cell 30?
No, this comes from the well. The well is well number 30.
But before it gets to the well, where does it come from in the sort that makes it cell 30?
It just comes from the tube. Just the,
So in other words, cell 30 is a random cell that's CXCR4 plus.
that was called the 30th cell. It was a 30th
So it's only
cell that was
reproducible in the sense of that one experiment, but you can't go back and recreate cell 30 with that gene expression pattern precisely.
It depends if cell 30 is completely identical to the cell that you're working with the second time. Hello.
I had a question about the last slide you presented. I mean, not the very last slide, but the last plot you presented as the average population. My question was, would it be important to do principal component analysis of those factors in order to get the most cell population autonomy shape, or would you want to have a lot of extra variables? So like you said, the variation averaged out when you have much of
I think in both cases it would be useful, because if you perform the principal component analysis, you'd be able to tell which ones made the most difference. And that would be helpful in its own right. But like you're also saying, the more you use, the less the more generalized the shape becomes, because you're touching on a fair bit of genes. But I think the idea at the end of the day is that you want to find out the genes that mainly characterize a particular cell type and use those genes to create these shapes. That's why the efforts that people have done in microarrays and sequencing is so important, because with those methods you can actually examine a whole host of genes and figure out the most important ones. Yes.
What's the minimum number of vectors you would need to get any sort of definitive shape?
That's a good question. 24, usually in my case. I'm only looking at 6 cell types, right? So I think as you start to expand that, you might want more. But the wheel, the pinwheel, has 24 strokes in here. There's no reason why you can't expand that to 96 or 248, 2048. This is just to make it simple. Yes, Brian?
So you've got this pinwheel, and you're looking at a couple of cells and you're comparing them. Is there a way to do that automatically in the software that will sort of identify the differences or how close they are?
Right, so this thing I just explained, you can code it into MATLAB. Basically, to say, take all the vectors that represent each one of the cells.
Does your GUI have that?
Yeah.
I was going to ask whether you're aware of anybody applying single cell mRNA, expression level analysis to immune cells stimulated in different ways, such as neutrophils or macrophages.
So Camille was about to ask me the question.
So you apply this to stem cells of various types from various sources. Are you aware of anybody looking at differentiated cells, but specifically white blood cells, human white blood cells, under different levels of stimulation?
The single cell gene expression experiments are still rather new. One of our faculty, Stephen Quaid, is responsible for the technology, and still in its inception stages. There are very few papers out on this. So no, I don't.
So you haven't seen anything like that?
I haven't personally, no.
Great. So we will have a party in the genetics library, and everybody's welcome to come at 11:30, 11:45.